as Neda said, yes, so what I'm going to present today is this theory that I have that system biology can help us understand what's happening in terms of adaptation of polygenic traits and how we can like bridge the vision of systems biology, quantitative genetics and population genetics. So to do that, I'm going to present you some results that date mostly back from what I did in my first postdoc with John Quackenbush on the regulatory networks, and some work that have been ongoing since then about heritability in those networks. And then I'm going to go to some opening about what it means for polygenic adaptation and some very preliminary results on the topic. But first, what I wanted to present was what I do right now in the lab at INRAE, which is the National Institute for Agriculture, Food and Environment. And I'm studying maize and its adaptation to environment. So as many of you might have noticed, there has been a series of very hard summer with a lot of drought. In particular, this was, so 2 summers ago in France, it was devastating for the yield for the major crops. And more generally, what we can see for all kind of different crops, I'm sorry, I'm still trying to manage the pointer, is that we have had a huge gain in yield this last 50 years due to mostly the progress in genetic selection. But the last 20 to 30 years, it's been kind of plateauing, and that's mainly due not to that we stopped making gains in terms of genetics, but that's the climate has been so unreliable in a way that the yield changed a lot from year to year. So we have reached here an impasse with the actual way of selecting crops. So what I do in the lab is to try to better understand the molecular basis of this crop's response to abiotic constraints and their adaptation to it, to try to help a bit improve the genomic prediction techniques. So I'm sorry. Yeah, so basically yield has many traits in plants, but also in Drosophila, in humans, in any kind of pluricellular organism, is a very complex trait with a genetic component up there, an environmental component, and a gene by environment interaction component. And I think that combining the information that we can get from the 3 different fields I've cited before will help us understand the whole picture. The second thing that's very important for how my research evolved in the last few years is that I came to realize after my PhD how important were the cis-regulatory elements and the gene expression regulation globally. So this is an example for maize. I'm going to show you later a similar picture that gather information from more organisms, but it's basically when you do a QTL or a genome-wide association study, when you look at the results for complex traits, and in particular developmental traits, for example, in maize, you can see that only 1/3 of the mutation falls within the gene bodies, and only 1/6 are in coding region. The rest is in introns. You also have about 1/3 of the mutations that are in promoters and 1/3 that are in intergenic regions, where the most likely suspects for having an impact on the phenotype are the distal cis-regulatory elements, such as the enhancers and the silencers. So taking this whole picture, regulatory mutations are likely to represent at least 2/3 of the mutations that impact the complex phenotypes for development in maize, at least. And it's often difficult to understand how exactly they impact the phenotype and how they can have an impact on adaptation. And I thought that maybe looking on the side of gene regulatory networks may help better understand this part. So that's what I'm doing in my lab today, is studying the role of cis regulatory elements and gene regulatory network in crops' response to the environment, with three main topics. The first is how those gene regulatory networks, how do they respond to the environment? The second is what is the role that they play into long-term adaptation? And the third is, can we use those gene regulatory network information to improve this yield prediction that has been quite bad in the context of climate change and environmental variation? So what I'm going to present today is this part, of course, my reflection that have led on this part that I'm starting to develop in the lab. Okay. So this is the outline. I presented it at the very beginning. So I'm going to first summarize a bit what I mean about the different field having complementary views of a molecular basis of polygenic traits. So the first view of a polygenic traits that you can think of, maybe, especially being a population geneticist for most of you, is the view that are in fact shared mostly with quantitative geneticists, which is that you have one polygenic trait that is determined by a number of loci that are independent. And often we use studies like genome-wide association studies or QTLs to identify them. We have many examples of such traits in many different organisms, plants, insects, mammals, yeast. Also. So what you are probably all familiar with is what it means in terms of adaptation at the genetic level, these models. So often we're working in the framework of the infinitesimal model, where there is a slight increase in frequency of many independent loci in time due to selection. And there has been an addition with, if you include some larger effect SNPs in the model, you may be confronted to what's called soft sweeps, which are pre-existing variants that are going to be selected and increase in frequency in the population. And the genetic architecture of the trait, whether it's a few SNPs that are participating to the trait, or if it's many, will influence how traits evolve and the kind of genetic signatures that you can expect for selection. The things remains that those molecular signatures that are not always easy to identify, in particular if you're working in natural populations and you cannot like follow the evolution like what a lot of you do with Drosophila and experimental evolution. And there is a necessity to better understand the genetic and the molecular architecture of complex traits to understand how they evolve. They have been evolving and how they may evolve in the future. Okay, so one of the drawback of this quantitative genetic approach is that mostly you don't know what's happening between the genotype layer and the trait, like often a global integrated phenotype that you observe. And this has been partly solved in the last few years by the inputs of two fields, systems biology and functional genomics, that have been looking at the effect of those SNPs on at the molecular level, and also systems biology that has tried to decipher how the different molecules were integrating at different levels from the genetics to the traits. And the more complex picture of a complex trait, sorry, I don't know what I think, yeah, okay, is, so you have many loci that are impacting somehow how the molecular processes are happening in a cell. And those processes are determining the sets of traits that are more or less related and that share more or less a genetic basis. So the first conclusion of that is that there is a level of pleiotropy in the architecture of complex trait. And with the question that comes immediately with that is, how does that level of complexity impact the possibilities for trait evolution? So there has been a series of work, and probably most of you have read about this omnigenic theory, that started with analyzing which pathway were involved in some polygenic traits in humans. So what they do was look at heritability, and they find as the proportion of variants explained by additive genetic values. And they looked at where was the heritability in the genes, and if those genes were expressed in tissue specific or broadly expressed in terms of fold enrichment and in terms of total heritability explained. And what they found was that it was the genes that were broadly expressed that that explained the most of the total heritability. From there, and some other data, including the results from systems biology, they built the omnigenic model. And so this omnigenic model, it states that you have some core genes that are acting on a phenotype, and that you have peripheral genes, and that's almost all the genes of the genome, that are impacting this trait in indirectly through the core genes. And one of the direct implications for that was, how do you evolve a polygenic trait if you have such a model in which basically everything impacts everything? So that was followed by another paper by Mathison, who expanded on this theory, saying that in the case of changing effect size depending on the environmental pressures and on the populations, this model meant that the effect size of the mutation would be changing depending on environmental pressures and population. So all of this together makes it, one, difficult to understand how one trait could evolve, and second, almost impossible to find molecular signature of polygenic selection for a given trait. So when I looked at these two papers, I was okay, I'm going to change work, do something else. My plan is dead. But then I looked again at the gene regulatory network, and those networks have some properties that can help explain why, despite those rather gloomy results for the concept of polygenic adaptation, polygenic adaptation is actually a possibility. It's found in nature, it's found in experimental evolution, so there is no reason why there is not a path on the molecular level for this adaptation. And I think the answer is in all of the properties that are hidden in those regulatory networks. So you have a lot of them. You have interactions between the genes, you have the redundancy of the networks, and you have some level of structure in the networks. And all of those three concepts will be very important for how a trait is determined and how it can evolve. So I'm going to show you some examples of what I just meant. So interaction is that generally in a pathway you have plenty of genes that are interacting. And this is a very nice example of a gene regulatory networks that regulates flowering time in maize. And you can see that, so the different colors are different populations. And you have had signals of selections at different genes in different populations that have all led to earlier flowering time of the plants, of the genotypes. So you can leverage this this quite level of interactions to select a few genes, not all of them, in all of the genotypes, and to select for a particular phenotype. The second is the redundancy, and this has been seen mostly at the interspecies level, but we know that it's very common during speciation to leverage the genome, the gene regulatory network duplication to neo-functionalize a whole part of a network. So it's here an example on the glucids metabolism in yeast, and there is a whole new metabolism of maltose and palatinose that have been that has arisen from the speciation and from the use of a duplicated gene regulatory network. So basically, this allows to build a new function or select a part of a new function of a functions in one part of the gene regulatory network, and keep the old one alive too, if it's a very necessary function. The third part that's important is that gene regulatory networks are structured in modules, and this is very important. So this is a characteristic that's present in all kind of biological networks that you can think of, whether here transcription factor gene networks, here that's an EQTL networks. You have structures, you have genes that talk more to each other, or regulatory regions and genes that are talking more to each other and not to the other. It's a bit like, that was one of John Quackenbush's favorite metaphor. It's a bit like your phone contact list. You have your family, you have your colleagues, you have your friends. You speak to everybody. There is very little chance that your grandmother is speaking to your colleague, unless it's a family business. So you can have elements that are connected through a network, but you can move one part without affecting the other part if there is not a lot of passes between the two different parts. So that's what I summarized here, is that you can evolve one function. They are, yes, connected to the other genes, but the fact that they are isolated in a module, it limits the impact of modification that you do in one module on all of these other functions. So there is one more layer that I wanted to add in this talk, is the fact that the network wiring is tissue or cell type specific. This is a result that I've seen in EQTL networks. This is a result that I have seen in transcription factor gene network. Here is an example on maize. I build a network for the seedlings, which are the very young plants, and for the husks, that are the mature leaves that are protecting the cob and the maize. And it's both leaf tissues, but what you can see is that they share, of course, a lot of modules in common. After all, they are leaves in a plant. But you can find tissue-specific modules that are activated or deactivated in the tissues, and that correspond to functions like here plant growth, or like here stress responses, or late stage leaf development, that are specific to one tissue. So this tissue specificity of wiring also open up an avenue to change the interaction in one specific cell type. So once all of this was, sorry, so this is a summary of what I just said as a quite lengthy introduction, was that those gene regulatory network properties, they allow space for evolution. The interactions in gene regulatory networks, they are at the source of pleiotropy, but they can also allow for convergent evolution using different pathways if needed. The redundancy can allow for neo-functionalization, and the structuration and tissue specificity can allow for evolution of one pathway without affecting too much the others. So my main question was, once we have all of this picture, is what can we understand about the molecular basis of complex trait and of their evolution by exploring the gene regulatory networks? That's where I'm going to switch to the two stories that I plan to tell you about today. So the first is identifying the role of regulatory SNPs, or which genes they are going to, which pathway they are going to affect using networks. Because one of the difficulty we ran into when we try to understand the evolution of a trait is that we have a SNP, we have the final function. Often the effect of the SNP is very, very small on the final phenotype, and we don't really know what the SNP is doing in terms of effect directly. So networks as a way to understand the function of like the impact of a mutation is the first thing I did in my postdocs with John Quackenbush. And I, for that, used EQTL networks. So I'm going to go a bit deeper on how I infer those networks and what does it mean. So first, let's go back to why I'm looking at EQTL networks. So I'm looking at it because of the importance of regulatory regions. This is the picture I showed you before. So it shows you that in cis, you have lots of possibilities of disrupting the expression of genes by mutations, and that often mutations that are associated to traits, they fall within those regions that have an impact in cis. You also have mutations that are in coding regions that can impact the gene expression in trans this time. So if you have a mutation in the sequence of a transcription factor, for example, or if you have, okay, it's not really coding, but it's microRNA coding. If you have a mutation in a microRNA, for example, all of this can impact the expression of genes in trans. So how do you access to this impact at the molecular level of regulatory mutations on the gene expression? You look at expression quantitative trait loci. That's the most immediate way to do that. I don't know how many have heard of expression quantitative trait loci before, but just to explain, it's you have one SNP that has two alleles here, for example, A and G, and they will impact the expression of a gene. If you have the A, you will express more the gene. If you have the J, you will express less. That's what you observe for these genes. An example in the blood of an EQTL for HLA, which is immunity self-characterization gene. So using this EQTL, well, the question is, how can we take into account all this complexity of interaction between regulatory regions and genes and the tissue specificity? My answer, of course, was by building networks. But to do that, I use data from the GTEx project. So the GTEx project is in humans, a huge project that gathers, as of today, 800 individuals, at the time 450. I used 13 tissues that are more than 200 samples per tissue. They have genotypes and they have RNA-seq data for about 30,000 genes. Those GTEx data, I use them to identify the cis and trans EQTL, so the EQTL that are affecting the genes directly on the same chromosomes and the one that are affecting the level of expression of the gene globally in both copies. And from those associations, I build a bipartite network with the SNPs on one side, the gene expression on the other side, and edges that were the significant association between SNPs and gene expression level. I did that for 13 different tissues, so I ended up with, 12 different tissues? 12 different tissues. And yes, so I ended up with 12 different networks in which I did network module detections and in which I found, so module detections is finding the mutations that are particularly associated to groups of genes and less associated to other groups of genes, basically. So it's dense zone of the network of like groups of SNPs and genes associated together. So that's what I did. I had 12 of them. And in the next question was, what does this module contains? And in fact, they contained either genes so that were clustered together the same way in all the tissues that I had, some that were shared between some tissues, and some that were a combination of genes that was tissue specific. So not very surprisingly, given the different expression, the different level of expression of genes between tissues, the module can be tissue specific or shared across tissue. As the first level of organization of the gene regulatory network, that's important to understand the effect of a mutation on a trait at the tissural level. So here are a few examples of the modules that I could find. For example, I had 3 muscle tissue: heart muscle, skeletal muscle, esophagus muscularis, which is smooth muscle. And in each of them, I found a tissue-specific module that was gathering genes that were specific of. Here you have striated muscle contraction, heart contraction. Here you have for the skeletal muscle, striated muscle cell differentiation. And here you have smooth muscle contraction. So you can see that you even at the level of differentiating 3 types of muscles, you can find tissue-specific modules. So this way you can find the genes that have a tissue-specific functions, and you can find the mutations that are impacting those genes. The second level that I was looking at was the more SNP-based level. In the network, you can define several levels of organization. So you have what's most commonly known that are global hubs or high-degree SNPs, that are SNPs that are very well connected through all of the network. You have leaves that are SNPs that are connected to 1 or 2 genes, not very connected. And you have the core SNPs. That was a notion that the Quackenbush team introduced, and which was a SNP that was very well connected within its module, but not connected outside of its module. So a local hub, if you wish. And the question was there a link between the position of those SNPs in the QTL network and their function? So I looked at the chromatin state, tissue-specific chromatin states that were underlying the core SNPs and the high-degree SNPs, so the local hubs and the global hubs. And I found that they were not located in the same regulatory elements. The core SNPs, they were more likely to be in active promoters and genic enhancers. And the high-degree SNPs, so the global hubs, they were more likely to be in intergenic active enhancers and polycomb repressed regions, which are often also intergenic enhancers that are not used in a tissue. So to summarize, the picture of this first study was that using this EQTL network, I could have one more step into the description of the effect of a SNP on a phenotype by having its effect on the molecular phenotype gene expression, by identifying the group of genes and whether it was tissue specific or on all tissue or in some tissues, and whether it was more likely to be an active enhancer, a global hub, or if it was more likely to affect only a handful of genes that were specific to a tissue and coding for a very tissue-specific function. So it was the first step of my study. The next step that I wanted to study, so this was the first part was the thing I really did during my postdoc. The second part is something that has been that I've worked on off since my postdoc, which is what's happening on traits heritability in a network. We have these nice properties of a network that can help us identify the role, but what about what we know when we know a heritability of a trait? How is it spread into the network? The SNPs that are associated in GWASs, or the SNPs that explain a part of the heritability, Where are they located? Are they in all of the modules, or are they clustered in some? Are they in specific positions in these networks? Those were questions that I wanted to answer for one specific reason, because it would help me identify which SNPs would be very important in terms of defining a trait. And if you have a trait for which you don't necessarily have you have an idea of which biological functions are involved, but you don't necessarily have a very good GWAS study or something like that, where should you look for in all the SNPs that you have to find candidates? So the first answer was provided by the study on cancer risk SNPs, but it was generalizable to all of the GWAS SNPs, was that there was a few, for each trait, there was a few modules that had many trait-associated SNPs, and a lot of modules that had only one or two. So basically, you had 2 to 6 communities per traits that were enriched in cancer risk SNPs for each network. So depending on the type of cancer, it determined the kind of the tissue-specific network. The second thing was, where are those GWAS associated SNPs? Are they everywhere in the network, the leaves? Are they local hubs? Are they global hubs? You could expect, say, well, global hubs this way, if they have a higher effect on the trait, maybe they are targeting lots of different genes. I tested for this hypothesis, and it happened that it was not the case. It was more enriched in intermediate degree SNPs. But then I looked at high core scores, so the local hubs, and then the enrichment was clear. So this is for all kind of GWAS, and this is for the cancers. We have a clear enrichment in local hubs for the GWAS associated SNPs. So SNPs associated to a trait seems to be at very specific location in a network that is clusterized in the local hub positions, affecting lots of genes in one biological functions, but not the genes outside of these biological functions. So here is a very important key of understanding how one SNP can affect a trait but not the other, is by its very specific position in the network, in articulating the network in a tissue-specific way, and at a very strategic position. So this is an example of such a SNP for cancer risk SNPs that I found in epithelial cell differentiation, epithelium development related module that was common to some tissues that had epidermal epithelium cells. And this SNPs was at the core of a module and linked to 5 different genes. One in cis that was known to be involved in metastasis, and 4 in trans that were all involved in extracellular matrix secretion. And this SNPs was known for breast cancer, in particular metastasis of association with metastasis of a breast cancer. So we have here a very nice example of what I was telling you before, a SNP in a strategic position affecting lots of genes in the same biological functions that is highly related to the traits. Beyond the SNPs that was significantly associated in GWAS, what can we say about the global heritability of a trait? So I started back with the GWAS data, this time taking all of the heritability results and projecting them in the network, because of course you have those that are significant, but you also have all of the others that are slightly associated, not necessarily significant at a genome-wide level, but that affect traits nonetheless. So the question was it true, those results for them too? So I redid this study with a bit more sophisticated tools that had been published and used in the way. in the meantime, and I did it with 10 different traits in a wide range with a wide range of genetic architecture. So it's all of the traits that I use. You have a genetic structure of them. So you have some oligogenic, like Alzheimer's disease, like type 2 diabetes. You have classic polygenic traits, and you also have quasi-omnigenic traits, which is height, adult size of the individual. The estimated genetic heritability also ranges from 25 to almost 90%. So you can see that it's a different genetic architecture. And I was wondering if the results that I was going to get were going to be different depending on the genetic architecture of a trait. I'm not going to maintain the suspense very long. The answer is no. I didn't find any difference depending on the genetic architecture. The global results were still the same. So I looked for heritability and found that this heritability was enriched mostly in a small proportions of modules, about 5 to 20%, depending on the disease. Of course, the most omnigenic or polygenic it was, the most modules you were going to find. So that was very logical. And the second thing that I found was that those heritability was enriched in modules that were making a lot of sense in terms of functions. So here, for example, you have HDL levels in the adipose visceral omentum module. So it's a tissue that's very important for the regulation of HDL level in blood. And you can see that this module is enriched for genes belonging to very low density lipoprotein particle clearance. This is one example, but I actually have many different examples for all of these traits and diseases across different tissues, depending on the traits, that are very compelling in terms of which modules are affected and where is most of the heritability located. So the picture that we have from this study is that the genetic heritability is concentrated in tissue-specific biologically relevant community within the network, and that most of the heritability is concentrated in a few modules that correspond to this description. So rather than having a picture of everything affect everything, you have a picture of mutations that affect the traits tend to have very specific location, tend to fall in very specific modules and be kind of with an effect enclosed into a modules that are biologically relevant. Same can be said for where is clustered the heritability. So the only slight surprise that I had was that in this study, when I took the whole heritability and not only the significant GWASs, I found that global hubs as well as local hubs were enriched for this heritability. So this was a bit surprising, but still it's there. So it was a very interesting result. So I'm going to do sort of a conclusion about that. So the conclusion is, if you want to look at where is polygenic trait heritability hidden in this EQTL network, you can see that it's not randomly spread. It's in tissue-specific or like a few tissue-specific modules with very specific functions, and it's in very specific locations in terms of SNPs, mostly local hubs, but also sometimes global hubs. So the structuration of tissue specificity of the networks, they help delineate which modules play a role in determining the polygenic traits. They limit the propagation of the effects of the mutations that can happen, and they open up paths for the complex trait to evolve in a context of everything is connected to everything. The node topology, whether they are local, global hubs, or leaves in these networks, it may help identify the loci that have a higher than average impact on gene expression, and is, sorry, there is a misspell, is it important to identify the selection targets? So that was my, that's the question I'm tackling right now, is the link between all of those properties that I've presented to you in the network and polygenic selection. So I'd like to finish by thanking everybody who participated to this study. So the GEVA team I'm a member of, so we are a collective of 6 researchers, a few technicians and engineers, and right now 3 PhD students and a number of Master 2 interns. I'd like to thank particularly Fanny Pouillet and Federico Sterlitz, that are 2 colleagues with whom I worked on the polygenic selection at a theoretical level. Rosanne Febe that did the first simulations, and John Quackenbush that thanks to whom I got my introduction into the world of gene regulatory networks. John Platik, who was my collaborator on the EQTL networks, and Catherine Stone, who jump-started the project on heritability 7 years ago as a bachelor student, and that I finally found the time to wrap up. So hopefully there will be a preprint soon. Christmas vacation kind of collided with my plans of getting the preprint out, and the publication afterwards. If you want to go further, these are the 3 papers that are summarizing everything that I've told you today. And I have a last slide, I think. That's a bit of an advertisement for me, as I just got fundings to develop this big project on maize adaptation to water deficit using gene regulatory networks. And I have a postdoc that's about studying the polygenic selection in gene regulatory networks, starting anytime from March 2024. So if you want to apply, feel free. Thank you for your attention.